with that, you can start.
I'll do that.
See.
All right. So first, thank you very much, Daniela, for that lovely introduction. So I took the liberty of changing the title a little bit as I realized that this is going to be more of a journal club kind of format. Um, I thought I'll just change it a little bit to spice it up a little bit. So I'll just be talking a little bit about the broad topic is novel approaches to study visual motor deficits in neurological disorders. And even though I'll be presenting most of the data that I've collected and that we published with stroke survivors, but I believe that these principles can still be applied to study other problems or other disorders as well. And also for this talk, I think I'm completely okay if you want to just stop me and ask me a question and interrupt. I feel very comfortable with that, so please feel free to do that. Though I'll try to finish my talking part in the allotted 25 minutes. So I want to first start with, for those of you who are probably not familiar with how visual motor coordination is studied, if you have a lower extremity background or if you're doing something more specific with imaging, most of the work that is done in visually guided reaching when you study this in the laboratory is basically what we call a center out reaching task, where whether on a virtual display, you basically ask people to fixate at a central point, and then you present a target in the periphery, and then the subjects basically make a rapid movement to that peripheral location. And what you can then do is you can measure reaction times, movement kinematics, you can find correlations. And this has basically been the fundamental way in which we have studied visual motor coordination in the lab over the last 4 or 5 decades. And I like to call this approach what we would call a serial information processing approach, in which I think from a motor system standpoint, what we do is we assume that the sensory system first has to sense, what changes have occurred in the environment. In this case, the target moving from a central to a more peripheral location, sense it, create a motor plan, and then simply execute that movement. But unfortunately, I mean, if you think about most real-world tasks that we do, or the ones which, when patients come and talk to clinicians, or when they, even those who have a disorder which causes very mild motor impairments, they would tell you that they have difficulties doing a lot of these activities of daily living, like driving, cooking, for example. And if you look at what really happens with the visual system and these kind of tasks, you see something like what you see on the screen right now. So on the left is basically an instance when people were collecting eye tracking data on healthy adults driving a car. And you can see that, they're constantly scanning the whole environment. They are, based on that continuous sensing, they're, changing, making slight adjustments to their actions that is allowing them to, operate this vehicle smoothly. And on the right is basically this figure from Mary Hayhoe and Donna Ballard, who show that, how do people basically, for example, fix a peanut butter sandwich. And you will see that, what these red lines here basically show saccades. The yellow areas show fixations. And what they've shown with tons and tons of studies is that in most of these activities that we do in our daily life, we're constantly sensing and acting. You see, you make a movement, then if you're trying to find something else, you scan the environment, you find it, and you make a movement. And if you think about it this way, it basically brings to question whether we should think about motor control within a more continuous framework, where where the actor, in this case, the human subject, is constantly sensing and interacting with the environment. Because what I'm trying to do, we're going to try to do today, is to make a case that actually I believe that this is perhaps a more difficult but a more rewarding and a more informative way of looking at disorders, especially neurological disorders, which do not cause the kind of significant motor impairment that we talk about, like spasticity, but mild impairments, which still cause enough difficulties in people's life that they have difficulty performing their activities of daily living. And so here's one a study that came out in 2015, where they published in Journal of Neurological Sciences, and they recruited stroke survivors who had very mild motor impairments. Um, and they still found that, if you put these stroke survivors on a driving simulator and give them real world situations, they would end up making errors that could put them in harm's way. And I feel like this is the kind of problem that, what my research and the kind of questions that I'm asking is focus on, which is these questions where the sensory and the motor systems have to continuously interact with each other. But unfortunately, if you look at what's like what's out there in the literature, and how we study motor control, this is a figure that I picked up from Candle and Schwartz, which is basically the way most of us at this point, or at least our textbooks tell us how we study motor control. That you have a stimulus, it creates a sense of perception. The perception feeds into the cognitive system, and the cognitive system basically creates a motor plan, and then all the motor system does is basically releases that plan into an action. And I feel that this way of thinking is in my opinion a little obsolete, for the lack of a better word. But it also not only just obsolete, but it also limits our understanding of how we should be studying, or we should be looking at impairments in the laboratory. So I'll give you one example, and this is again one of many studies. This is actually done by one of my friends and close collaborators, Jennifer Semler. She's at the University of Delaware, and she published this study in neuro rehab and neuro repair, where so on the top you see there's a Kinon robot in which which basically supports people's arm, and then in an augmented reality environment they make different kinds of arm movements. And so what on the top you'll see is the center out reaching movements, the kind of example that I just showed, for healthy control, with both the left and the right arm. And you'll see that, movement trajectories are fairly linear. If you look at the kinematics, they're fairly bell-shaped, which is what we expect most movements to look like. But when you look at a stroke survivor, so this is a stroke survivor with a unilateral deficit. On the left is is the impaired arm, the contralesional arm, and you can clearly see very strong deficits there. I mean, it's very apparent. But when you look at the other arm, the ipsilesional arm, or what we would call typically a less affected arm, that's where you would see that perhaps it looks very similar in terms of its kinematic characteristics to that of a healthy control, both in terms of the spatiotemporal trajectories as well as the kinematic. They look very similar to what you would see in a control subject. And that would give us the impression that maybe the ipsilesional arm is fairly intact. It's good. It can go about performing daily activities of daily living fairly well. But if you put this person in a different task, so this task that I'm going to show you is called the trails test. And for those of you who work with clinical populations, you probably know this task very well. But for those of you who don't, I'll just repeat this very quickly. So this task is basically a task which is done with a paper and pencil and a stopwatch in the clinic. And it has two variants, Trails A and Trails B. In Trails A, people connect numbers 1 to 25 in a sequence. So they just take these lines and connect them. Trails B is a slightly more challenging alphanumeric variant in which they have to switch between numbers and letters. So they go 1A, 2B, 3C, and so on. And they have to connect those dots in the shortest possible time. And that's basically the instruction that's given to them. And so if you see this test being performed, so on the left is a healthy control. And on the right, this is the Trails B test, or TMTB, as we call it. And on the right is a stroke survivor who's doing this test with their less affected arm. And I hope you can see that this stroke survivor clearly, they took about 4 times longer than this control. And again, this was a stroke survivor who had very mild motor impairments, but they're still struggling to do this test. And that's when these kind of difficulties come out in the open, clearly that you can see that there's something really not right with how they're going about doing this test. In fact, the green crosshair that you see is their eye movement. So we're tracking their eye movements in this case. And you can see clearly they're struggling to find the next target. They can't keep track of what was the next number or sequence that they have to find. And that obviously creates difficulties in performing this test. Now, this test also correlates very well, by the way, with driving performance, and there's a reason why it has become a very popular test. In fact, I believe that there are some states that do require people who have suffered any kind of a cerebral injury to actually take this test as well before they take the road test. Um, I'm not sure which those states are, but I know that in some countries, they do require people to take this test before they can take their driving test back if they've suffered a cerebral injury. So the point that I just want to make so far from what I've covered is that perhaps this serial information processing framework, in which we separate the sensory, cognitive, and motor systems, is perhaps not optimal if we want to study neurological disorders on a continuum. And this is an approach like a continuous motor task, which can perhaps be more informative. And that's basically the kind of evidence that I will try to present today. So for this study that we did, we asked people, different groups of controls and stroke survivors. We had 37 young controls, 16 older controls, and 16 stroke survivors. And we published the study, so you can find all more information if you're interested in the clinical characteristics in this 2017 paper. But we brought them in, they did a bunch of different tests. And one of the tests that they all did was this trails test, both A and B, on the skin arm robot, where we basically tracked both their limb movements and eye movements simultaneously. And just to give you a very quick overview, what we found was what you would expect, that the total time taken by the stroke survivors was much, much longer on both Trails A, which is shown in green, and Trails B, which is shown here in gray. They took much longer compared to their aged equivalent controls. So this
Rakesh, can I interrupt you briefly? A quick question here.
No, please.
Just a side note, the difference between the B and the A, the times for the B and the A, that difference time is supposed to be correlated with a component of executive function. Mm-hmm. Did you look at that? So maybe deficits in the motor capacity versus cognitive deficits that stroke patients may suffer from?
Yeah, so just give me a few minutes, I'll talk about all of that. Absolutely. So the main difference that Satya is talking about is in that when you're switching from numbers to letters, yes, it does involve the executive component, what we would call these prefrontal higher order executive functions. And I'll show you those results in a second. But to just to summarize what I have so far is that yes, stroke survivors take much longer on both the simpler variant as well as the more complicated variant. And that is the kind of information you could also have gotten from a simple paper and pencil version of this test. But what robotics and eye tracking allows you to do is to basically dig a little bit deeper and figure out how these, what you would call visual cognitive deficits are basically impacting motor performance in this test. So by putting this test on the robot, we make this test both a visual cognitive test as well as a motor test, because they have to make these large reaching movements across the entire workspace instead of just, making these small movements on a piece of paper. So let me start by first telling a little bit of story about the eye movements first. And so what we did was we looked at different aspects of their eye movements. And I'm going to go in the opposite order, so from right to left. So the first variable that I show here is the oculomotor variable. And what we measured here was basically the average saccadic time, how long a saccade. So saccades typically last anywhere from 20 to 100 milliseconds. And we first wanted to ensure that it was not, if there are any differences that we see in their performance, if they were because of this, because of deficits in oculomotor function or saccadic speed. And what we found were two things. One was that first, the average saccadic duration, which is, so here, the black circles are basically controls and the gray circles here are stroke survivors. And you obviously, on the x-axis here is saccadic duration. On the y-axis is the total time that they took in seconds to complete the test. And what we found was basically no correlation between the oculomotor variable, which was saccadic time that they took, and the total time to complete the test. And there were also no differences between the three groups, the young, older, and stroke survivors, they all had similar circadian durations, which was an average of about 50 to 70 milliseconds, for example, between these groups. The second variable that we looked at was visual perception, which is that once subjects look at something and they're trying to find, for example, say the letter D, how long does it take them to recognize that what they are looking at right now is the letter D, or if it's not the letter D? And so again, we did the same thing. We looked at the average fixation duration, and we correlated that with the overall time taken to complete the test. And we found, one, that there was no correlation between this and the total and the overall performance, as well as there were no differences between the groups. They were all pretty much similar in terms of the average fixation time, which was roughly around 325 to 350 milliseconds. But the final variable that we looked at was how many number of saccades or fixations were made by these different groups. And that's where we found a significant difference between the stroke survivors and controls, or their age-matched controls. And we also found almost a perfect correlation between that and the overall time that it took them to complete the test. So very strongly correlated. So clearly that told us that the way in which they are going about trying to find where the object is in the workspace is perhaps the real problem here that we need to probably dig a little bit deeper into. Now, before I tell you about what we did next with this data, about why stroke survivors were taking so much longer, let me just give you an example of how visual search is typically done. And for those of you, again, who are not familiar with the visual search literature, it's a very, they've done a lot of very good work, and they have very established mechanisms of how we humans go about doing visual search. Now, but to summarize for the context of this discussion, I'm just going to say, let's take these two examples where we're trying to find, say, a loaf of bread. And I can present to you the loaf of bread in two different ways. The first one's on the right, where the loaf of bread is amongst a random category of objects, and I ask you to find that bread. Now, in this case, most people would be very similar because there's no rule-based way to search it. So you'll randomly search wherever you feel like, and then once you found your object, you'll see this is where the loaf of bread is, which is right about here. But in a space like a kitchen, which has a contextual relevance, most of us who don't have cerebral injuries, when we, and I don't mean that in a disrespectful way, what I do mean is that we use some prior knowledge to basically search in a very structured way. And what I will show you is that this is what is basically missing in the stroke survivors that we studied previously. So for example, if I'm looking for a loaf of bread in a kitchen, I would first start my search on flat surfaces. And once I've exhausted all the flat surfaces, will I look at something where, there's little or no probability, like a vertical wall or something, or a microwave. So when we do a task like this in like the trails test, one thing that we found was that most healthy people, young as well as older people, controls, they come with some prior understanding of the world of how they should do visual search, and they apply that understanding into this test. And how does that play out? So there are three ways in which we thought visual search can be done by people. One is what we call topographic search, which is that, let's imagine that your hand is on this number 15, and you're looking at the next target in Trails A, which is 16. 16 is going to be actually very close to where 15 is. So it's going to be in a very close neighboring proximity to 15. So most young adults would basically restrict their search in a very small area right next to where their hand is, because that's where the probability of the next target being available is highest. And then you could basically search in a very small radius or somewhat larger radius, but there's no point looking for something in the opposite end of the workspace, because that's where the likelihood is minimal. And we call this strategy topographic search, which is a spatial rule to search in a rule-based fashion. The next is working memory, which is something which I guess most of you are familiar with. It's a short-term memory, which is primarily mediated by dorsolateral prefrontal processes. And in this case, what happens is that when you look at something, so let's just say your hand is again at 15, and you're looking for this number 16, but you make a fixation at 8, 18, or 14, you keep that in your short-term memory, so you're not wasting time going back and looking at the same number again. And most people, again, healthy adults, would basically use their working memory in some capacity to basically guide their visual search and not waste time going looking back and forth between what they've already looked. And then the third thing that we looked at was that when you are doing this test, whether it's a paper and pencil based test or whether you're doing it on a computer, when you have when your hand has traversed a target in the sequence, we leave that pencil mark on the augmented reality display. So which means that if you're at 15, this is what you're going to see in the workspace. So if you're looking for 16 and you've already basically covered this area, you shouldn't have any reason to look in this space, but focus your search on those areas where your hand has not been. So we thought about this, we modeled these three processes, and what we found was that most people, including healthy adults, they did not use this third process. Most people use this first and the second, working memory, as well as a topographic rule-based search. And that's how they conducted their search. Now, what we did next was, I think I'm not going to go into that because again, this is all available. It's published, the model and everything has been published. But I'm going to give you a brief summary of what we found in terms of relevance. So what we really found was affecting why stroke survivors were making so many more saccades than age-matched controls. So we took the data for each subject. We put it through a mathematical model. That's basically a figure from the paper. But here's what we found. So this is a three-dimensional figure where this x-axis is the log of total time taken to complete the test. The y-axis is basically a measure of working memory, which is how many things that they kept in their working memory at a given time. So for example, number of 2 would tell you that they were keeping about approximately 2 things in their working memory. Now, exactly how this correlates to real world, you will have to read the paper, but I'll just give you this broad number that just the higher the working memory, the more the number of items you're storing, which means that the longer you take before you look back at the same number that you again. And the final thing here is the TS, which is the topographic search, which is the smaller this radius, the smaller this number, the larger the radius. So if you're randomly searching all over the space, then your number would be very high, like 20 or 25. Whereas if you're restricting your search in a smaller area, you'd be in a very small space. Let me change this, sorry. So what we found was we basically then put everything to this model and then used cluster analysis, which is a machine learning technique to just divide our sample participants into different groups. And here's what we found. So here triangles are basically the controls, the age younger and older controls clubbed into one group. And the circles here are the stroke survivors. And we found 3 different groups in which we could categorize them broadly based on what we got from the cluster analysis. The first group was this control group, which in this group, most of the people were controls. And what we found was that they basically used working memory, but they also, searched within much larger spaces. So they would use their working memory effectively, but search in a larger volume. Whereas this group, the red group, which is kind of hidden behind, sorry. This group was also consisted mostly of controls. This group did not use working memory at all, but they restricted their search in a very small area right next to where the hand was. And when you look at the performance of these 2 groups, the green and the red groups, they were pretty similar in terms of the overall time that they took to complete the test. Whereas the group, blue group, which consisted mostly of survivors, the circles, this group did not use working memory, but conducted the search in a much larger area. And so more randomly, without memorizing or keeping in mind where, what numbers they had previously looked at. And this is what caused them to make many more saccades and probably contributed to their longer time to complete the test. So the next question, which I'm sure all of you are interested in, yes, it makes sense, but does it actually affect their motor performance? Because yeah, we get that there are cognitive impairments which are causing them to make more saccades, not remember what they've looked at, but does it affect motor performance? And this is what we addressed in this 2018 paper, which I guess some of you may have read, but we tried to see if there were any relationships between these saccades that they made, these excessive saccades, and these movements they were making. So here on the top, I'm showing movement kinematics from two of the participants. One is a control participant on the left. And you can see that in this case, their movements are fairly bell-shaped, what you would typically expect in a center of reaching task. Whereas when you look at a stroke survivor, you see that they take much longer, their movements are slower, whereas they also have more what I would call bumpier movements, which means that, if you see this, they make more, there are more of these peaks in their movements compared to controls. So what we wanted to see was, is there a relationship between these saccades that they are making, excessive number of saccades, and these excessive, peaks that we see in their limb movements? And if that is in any way also causally or not, but in some way contributing to their slower movement speeds. So indeed, their movement speeds were slower, which is what I'm showing over here, that the stroke survivors made slower movements than control. And then what we did was we looked at these two, their eye movements and their hand movements together. And that's what I'm showing in this plot over here. The x-axis is time. The black here is basically the limb movement of between two adjacent targets, say number 15 to 16, which is shown here in black. These peaks here are highlighted with these triangles. And then these are the saccades that are shown here in the gray. Saccades basically, so these are all the saccades that you see over here. So what we did was we basically took every single peak that we found in the limb movement, and we basically looked at about a 400 millisecond window prior to and after that peak. And you may ask, why 400 milliseconds? So we thought that if these saccades are in some way contributing to these jerkier movements, we need to have a window which basically covers the window of a standard reaction time, which would be 250 to 300 milliseconds in a task, in a center of reaching task. We went a little bit conservative. We went to 400 to cover that. And what we found was that there were 4 times as many saccades in this window as there were in the window after every peak in the hand movement, which suggested to us that perhaps these excessive saccades are in some way, we don't have causal evidence, but we have correlational evidence to show that they were probably contributing to these jerkier as well as slower hand movements. And that's what I'm showing over here. These are on the x-axis, here is saccades that people made between every reaching movement. And here is the number of peaks per reach, as well as the reach speed. And what we found was that they were both very strongly correlated with the number of saccades that people were making per reaching movement. So between every consecutive reach. So the take-home message is that stroke survivors made many more saccades that likely slowed down limb movements. Again, this is not this is a study which we can get us to causality between those two phenomena, but it at least lays the correlational framework for us to study that further. And recently, when we published that study, I thought that at that time I thought that we were probably one of the only labs that was looking at something like this. And I could not find any other studies that had shown something similar. But obviously, since then I've seen that other people have published similar results as well. This is one study that I came across recently, which was published in the Frontiers in Neurology, done by J.R. Rizzo, who's at New York University. And what they showed was, they asked people to make these kind of center out visually, reaching movements, visually guided movements where the subjects were making this movement on a tabletop, but the target was actually presented on a computer screen. So they had to map this computer monitor onto this space and make these reaching movements. And what they found was something very similar. So here, these dots basically indicate gaze location. And on the top is, this is a control subject, this is a stroke survivor. And you'll see that in the case of the control subject, they make fixation, then they make a move, they basically make a saccade, which basically kind of is made around the same time as the reaching movement. and then once they've landed their hand on the target, then their gaze is fixated where the target is. Whereas when you look at a stroke survivor, you'll see that even before the movement is initiated, they are still, looking. They're making saccades before, which is not at where their hand is. They're making saccades while the hand is still moving, and this is in some way also supporting what we've found and what we showed, that stroke survivors are making these unnecessary saccades during a limb movement, and maybe it is actually contributing to uh, slower limb movements as well. Again, this is just population-based result that they're showing, that they actually showed that stroke survivors made many more saccades and task-irrelevant saccades in their study, both before as well as while the limb movement was already underway. So this is just an overall picture of where of some behavioral description of what we've found, we've observed in these stroke survivors who have very little motor impairments, but who still exhibit these strong visual motor deficits that kind of play out the way in which I've shown in these last two studies. And I think I'm going to leave this for discussion because right now we're thinking about how to think about neural mechanisms, how we should be studying this problem, defining this problem and studying this problem in a structured way to get to the bottom of this. So if we come back, maybe I'll come back to this, this particular slide that I have on circuits. But I'll just end here. Obviously, I just started my lab here at Penn State last year, so it's pretty small. I have one grad student, and these are all my collaborators and colleagues, both at my current institution, my institution, and some across the globe in France. So I want to thank all of them for all this work, because all of what I do is made possible with all this great interactions and team that I work with. And also, I'll be a little bit shameless and advertise that I'm actually looking for a postdoc for my lab. And anybody who's interested in using experimental and computational approaches to study visual motor control in healthy and adults as well as clinical populations is welcome to reach out to me. So from that, I'll just rest it. Thank you.
Okay, thanks, Darkesh. Yeah, so now we're gonna do the breakout rooms and to have the small groups discussion, and then return to the big groups. Okay. So if something pops up on your screen, like, you can just click OK.
If you take a bunch of healthy controls and you have them do an easy visual motor task versus one that's more challenging, I don't know exactly how you would increase the difficulty, but does the number of saccades change as a function of the difficulty of the task? And I guess you could vary the cognitive difficulty versus the motor difficulty if you can find ways to dissociate the two. But
That's a great question.
Basically, Darkesh, you can put a whole bunch of distractors and vary it.
Right. So I think I get your question, and I think so. Here's my response to that is two. So one idea that in the traditional motor control world, there's a phenomenon that we call gaze anchoring, which is that in these center out tasks, what people would do is that they would basically land their saccade to the next target and then anchor their gaze till their hand reaches that. What you're suggesting is that what if we put distractors while this movement is underway? And I think that is a great way to probe. And that was the neural circuit slide that I didn't get a chance to talk about, is that there is a law in the visual, in the field of visual sciences, cognitive sciences, and they call it the Bloch's law, which is that the luminance of a target, for example, its intensity and the time over which that is presented basically dictates how much energy lands onto your retina. And during the course of development, we figure out how to basically suppress that, like, for example, I'm looking at a screen right now, and there are 10 different things competing for attention at the same time. But I'm suppressing my urge to land a saccade there because I've figured out a way to not let those, those nerve impulses grab my attention. But if it's possible that some of these subcortical pathways, especially those retinotectal pathways, if they are hyperactivated or if there is some issue there, then you're right. There could absolutely be an issue that some stroke survivors may have a higher propensity to just look at anything that even demands a little bit of their visual attention. Because like you probably that better than I do, but in the superior colliculus, the visual and motor maps are exactly in register. So if the sensory layers in the superior colliculus are hyperactivated, they may actually cause people to make these random unnecessary saccades. And in a task like simple center outreaching, there may not be a huge cost to it. But when you are driving in a busy place like New York City or in a busy environment, and you're randomly getting distracted with anything that's grabbing your attention, that could have a real world cost. And I think that's what we're planning to study a little bit more systematically, to figure out exactly, is this a way in which we can probe how these subcortical pathways are basically involved in attentional control in these kind of continuous visual motor tasks. If that answers your...
Yeah, thank you.
Rakesh, I had a question about whether the mechanism of the saccade during the movement versus dwelling, when you're doing the visual search, if you can comment on the different mechanisms for that, or if you think it's like they have the same source.
No, you're right. That's a great question. So I think what Daniel is asking is that when their hand is resting and they're searching for the next target, right? If those saccades are any different than what they're making when their hand movement is already underway, right? Is that
yes.
Okay, yeah. So the short answer is that yes, there are some differences there. What we found is that most people, controls as well as stroke survivors, both those groups made saccades at both times, when their hand was resting as well as when their hand was already underway. The When I showed you those, that plot where I showed you that we looked in the 4 millisecond window. So we were only looking at those saccades which were being made when a movement was underway, because we were looking at the probability of a saccade happening around each, movement peak velocity.
Mm.
And what we found was that that's where there were differences between, those two groups. There were more saccades leading up to a limb movement peak compared to after that.
Mm-hmm.
So I was primarily focusing on what they were doing when a limb movement was underway, not when they were actually, dwelling. And that's basically what we've been trying to show in that second neuro rehab and neural repair paper. Yeah, that's a
So
very...
just had a follow-up question to that. That's a really good point. So you talked about gaze anchoring. So why do you think gaze anchoring occurs if in your studies you see that people make saccades while the hand is moving, whereas in these earlier papers, I think it's Negers and Beckering, where they show this typical gaze anchoring, although there's only one target that they need to move to.
Right.
Yeah, just wondering why that happens with a single target and maybe not multiple targets.
So that's a great question, Christina. And I can just tell you that from what I believe it's an artifact of the center outreaching paradigm. The task, the paper, if you look at the slide, the Mary Hayhoe and Dana Ballard paper, 2005, Trends in Cognitive Sciences, they basically put eye tracker in a real world task when people were making a peanut butter sandwich, sorry, a peanut sandwich. That would be hard to make. But and you'll see that their gaze is constantly moving. There's nothing like gaze anchoring in that data set that they presented. And if you think about any real world task, when do we actually anchor our gaze? Like even when I'm drinking, like talking to you and trying to get this, get a sip of water, I'm not doing, I mean, that would be weird that, I'll say, oh, Christina, just hang on. And then start talking. I mean, that's not how we do stuff. So I believe that the gaze anchoring phenomena that you're talking about, to me, is an artifact of this serial information processing framework in which we just have one target to move to. What else would you do? Because there's only one thing grabbing your attention. But if you have, in a real world where there are multiple objects that could, try to attract your attention, then I really don't think that there would be something like gaze anchoring. So I don't know if that convinces you.
Yeah, that was great. Thank you.
Mhm. Okay, so we're like at 1 o'clock right now, and I want to thank again for everyone to be here. And our next meeting will be on the 25th, and the speaker will be Dr. Michael Borick from Emory, and we will discuss the topic augmenting plasticity to enhance motor learning. And those who would like to speak more with Tarkesh are welcome to stay. Mhm.
Tarkesh, I had a question for you.
Mhm.
So let's say that there is some, I'll just extrapolate broadly by saying, let's say there is some causal relationship between eye movements and arm motion, right? Saccadic movements and arm motion. From a rehabilitation perspective, does that suggest that we can manipulate, the efficacy of arm motion through visual training, right? Training saccades and such. This was a topic that came up in our discussion room, which we were kind of wondering about.
Right. That's a great question. I am going to direct you to a paper. Give me 1 second. In fact, there are multiple clinical trials already going on right now, or some of them have concluded. Some of them, I guess, at least one of them was ongoing the last time I checked. But I'll give you. So there's one paper, Neuro Rehab and Neuro Repair, 2013. The first author is Kerkhoff, Kerkhoff. The title of the paper is Smooth Pursuit Eye Movement Training Promotes Recovery from Auditory and Visual Neglect, a randomized control study. And in fact, there is, if you send me an email, I can send you a couple of other papers that there's a review paper that I recently read where uh, people have been they're trying to use eye movements in ways in which, I mean, hoping that it'll basically have some kind of an, effect on arm movements post-stroke or other disorders. So there's a bunch of such trials that have concluded already. And this guy, George Kerkhoff, he's published another paper on which is titled Smooth Pursuit Bedside Training Reduces Disability and Unawareness During Activities of Daily Living and Neglect, a randomized controlled trial. Also in Neuro Rehab and Neuro Repair 2014. So these are some of the things that you can take a look at where people have been trying to look at these issues and doing clinical trials. So the idea that eye movements or training eye movements can do something to can change limb movements to me, is not sci-fi. I mean, you can trace neural pathways and say, yes, that you can induce plasticity in these networks with training. How to do it in a very controlled, systematic way, in a target way, is a difficult question to answer. But at least what these two studies that they've published have shown that there's at least some evidence to show. But again, let's come back to the point that, we do not know much about how the visual and the limb systems interact. I mean, we know quite a bit, but again, most of what we know is within a serial information processing framework, which again has its own limitations, gives you a certain amount of information. But if you want to think about functional improvements, then there's got to be a more systematic way in which we can study this and address this and figure out how eye movements can induce, improvements in limb motor function.
Can I just jump in there for a second on the neglect aspect of things?
Mhm.
So I think that the smooth pursuit training, first, the benefit that those patients got had to do with a reduction of the impairment in their visual spatial attention deficit. So I think that that's a pretty specific type of scenario or disorder that is different from, I think, the more general types of motor control issues that we've been talking about.
Yeah.
I think it's more akin to the type of effect or benefit that patients with hemispatial neglect get after prism adaptation therapy treatment.
Yeah.
Which we still don't really know exactly how that works either. But I think what this conversation does bring us back to is some aspects of what we were talking about when we were discussing the optimal theory of motor learning. Now, if you could control someone's eye movements, you would also almost unavoidably be controlling their focus of attention. And I think we've talked a number of times about how that affects both learning and online performance. So I think that if you're going to, I think that I think that we already have plenty of evidence in healthy people, elite athletes, among others, that where people are looking has a significant impact on their motor performance.
Right, right. So I think that there's another.
I was just going to say, as a
sorry.
therapist.
Sorry, Rachel, I think I didn't really catch what you said.
Oh, I was going to say, like, can you hear me now?
I can, yeah.
Yeah.
I can, yeah.
I was saying that I think in therapy, oh, I think my internet's bad, but in therapy, like, I think oops, am I back?
You're back, yeah.
Okay. I think in therapy, there's the underlying idea that besides hemispatial neglect, that like in concussion rehab and vestibular rehab, movements are being used a lot as a target for treatment, but I don't think that's made its way into stroke as much. I think those areas more in the vestibular side of therapy are kind of leading the way for how we could potentially do more eye movement training, but I think there's the gap then in what that would mean for stroke.
Right, and I think that you're making the same point. So the point that Alex made is that that these studies, that these clinical trials that have been done, they're still on the visuospatial, side of addressing visuospatial side of the problem. And the point that I'm making, that I want to make, is that at least there are two studies that I can talk about, which again, these are basic science studies that were done. One was done by Luc Sellen. He's somewhere, I think, in Belgium or Holland. And what they did was very interesting. So what they were doing was this motion coherence task. So they were looking at these random dots moving left and right, and subjects had to decide whether the dots were moving left or right. And what they found was that with that, the amount of time that the subjects were basically looking at that, those dots moving left or right, there was a continuous signal that was basically being accessed by the limb motor system, because so what they did was they perturbed the arm during that time when the subjects were looking at those dots. And what they found was that the gain of the long latency reflex basically scaled with the time. And they used that to show, look, this is a very good proof that, that the limb motor system is continuously accessing these motion signals, which are basically being processed by this medial temporal area. Or at least that's what we know from the monkey literature. Now, where I kind of really got excited with that study was that this medial temporal area that we know from monkey work that's involved in motion perception, this is also a key node in these pursuit eye movements. And that's where they significantly diverge from saccades, because this area does involve the pursuit eye movements. So when I saw these kind of studies, like these clinical trials trying to engage the pursuit eye movements, I wasn't surprised. I was like, this makes sense. That we know that through this area, there is a direct channel that the oculomotor system has to the limb motor networks. How can that be harnessed? How can that be studied to improve function and whatnot? Those are the questions which I think are difficult, incredibly challenging. But I still think that it's a way in which, if we have to systematically study these links, that might be one point where we can focus on, motion, pursue. So the point I want to just reel back and say is that I think that there's more that can be done with pursuit eye movement training, but we first should do some basic science work to see how is this network engaging with the limb motor system. That would be my 2 cents, I think. Yeah.
anybody has any more comments or questions for Cash. No. So
well, thank you again, both of for you. It was really nice to come here and talk about it, and very glad for this opportunity.
Thank you.
And see you guys soon. Thank you.
Thanks.
Bye.
Bye.